and today we will be talking about mitochondrial defects arise from nucleoside or nucleotide reverse transcriptase inhibitors in neurons, potential contribution to HIV-associated neurocognitive disorders. I chose this topic simply because HIV is a really relevant field of study in our careers, and knowing how our treatment plans can lead to potential harmful side effects is essential. So we know that the major factor in neurocognitive disorders is the active HIV infection in the central nervous system tissues, and that halting viral replication is essential to preventing further degradation of these essential systems. CART, which is combined antiretroviral therapy, where you use multiple different antiretroviral drugs, which will inhibit the viral replication life cycle at different stages, has drastically increased the survival rates while preventing severe HIV-associated dementia, but unfortunately it has not reduced the overall prevalence of HIV-associated neurocognitive disorders, or also known as HAND. Although it has not been studied clinically nor in the lab, there seems to be a potential for these treatments to contribute to these disorders. So in CART, NRTIs, which are nucleoside or nucleotide reverse transcriptase inhibitors, are a class of drugs which are essential to these regimens. But they've noticed that long-term treatment using the NRTIs has severe side effects such as myopathy, lactic acidosis, peripheral neuropathy. And they have come to the conclusion that these are all related to mitochondrial toxicity, which is integral to many types of neurodegeneration. So these harmful side effects are the result of toxicity resulting from the NRTI drugs, and this is mediated through the inhibition of the mitochondrial DNA polymerase, specifically Pol gamma. This is due to the fact that it causes a reduction in mitochondrial DNA synthesis, a reduction of mitochondrial content, and these two combined lead to various mitochondrial dysfunctions, such as diminished ATP production, as well as an accumulation of the orphan respiratory complex subunits. So using this knowledge, they came up with the primary goal for the study to determine whether or not NRTIs produce mitochondrial dysregulation and toxicity in neurons. So in the course of the study, they used 4 different NRTIs, which are currently being used or are under research right now, and these are DDI, AZT, TDF, and FTC. Using these 4 NRTIs, they treated the primary cortical neuronal cultures, such as seen in this figure, and they also used a previously established panel of endpoints to assess the mitochondrial DNA levels, the oxidative phosphorylation activity, retrograde signaling pathways, and mitochondrial homeostasis disruptions for each molecule. This allowed them to have somewhat of a control group to determine the implications of each of their results. So these 5 results were obtained in the course of the study, but I'm going to specifically focus on these first 2, which were the results of dose response and time course of NRTI toxicity. And the second one being DDI prevents mitochondrial DNA synthesis and diminishes mitochondrial DNA content and mRNA expression. So the first one, they were looking at the viability of the cells with an MTT assay, which is where it's a colorimetric assay to determine the metabolic activity of the cells. So the first part of this figure is looking at the viability of the cells when treated with 1 micromolar, 10 micromolar, and 100 micromolar of each of the 4 NRTIs. These studies were done over the course of 14 days, and using PBS controls, they looked at the viability. As you can see, as right around day 5, the cellular viability of all 4 cultures were similar to the control cultures, but as the progression went on to day 14, they found that cellular viability was greatly decreased. They also used immunocytochemical staining to look at how the cellular structures of the neurons were affected at day 5. So using the neuronal cytoskeleton marker MAP2, they looked at the structure of all of the cell neurons. So the first one is the control with PBS, and then as you can see at day 5, all the neurons treated with the NRTIs appeared to be similar to the control. They also used a presynaptic marker, synaptophysin, to look at the synapses of the neurons. So same as before, PBS and then the 4 NRTIs. The red is showing that they were fairly unaffected with the treatment after 5 days. They also concluded that DDI prevents mitochondrial synthesis and diminishes mitochondrial DNA content and mRNA expression. DDI stands for dianosine, and they determined these conclusions based off of using a BRDU stain to label the newly synthesized mitochondrial DNA. As you can see right here, after they were treated, they saw a large reduction in the mitochondrial DNA using the DDI, while the other 3 NRTIs were less affected. They also used real-time PCR to measure the amount of mitochondrial DNA content. And as you see, right around day 1, it seems fairly unaffected, but the real difference lies in the fact that DDI at day 5 greatly reduces the amount of mitochondrial DNA when compared to the PBS control group. So they concluded that although NRTIs are highly effective in preventing HIV proliferation, due to the fact that they are able to penetrate the blood-brain barrier, they have dual potentials. They have the potential to protect from, and they also have the potential to contribute to the development of the HIV-associated neurocognitive disorders. There are 8 currently or previously licensed NRTIs, such as AZT, which was in the study, DDI, DDC, D4T, 3TC, ABC, TDF, and FTC. These first 4, which are in bold, exhibit more off-target toxicity, and as a result, their clinical use has been greatly reduced. They also concluded that while it contributes to the development of these disorders, mitochondrial DNA depletion is not the sole cause for it, for producing abnormal mitochondrial functioning in neurons. They conclude that it is likely due to the fact that the deficiencies in the mitochondrial DNA encoded proteins are the key drivers for these induced respiratory defects in the mitochondria. And they stated that they need to further this study for a longer time period to produce overt mitochondrial toxicity. Future research is to see how these NRTIs with the potential to contribute to these disorders how it extends over a period of a longer amount of time. And they also want to see other NRTIs and their effects on it. And that's my reference.